Okay, so welcome to this next video in the playlist on inflammation and angiogenesis. In this video, what we're going to do is talk about the different types of capillaries. Okay, so the structure for this video is we'll start by talking about what capillaries are. So we'll look at them in the context of the larger picture of the circulatory system. We'll then look at the three main different types of capillaries, and finally we'll talk about how you can transport molecules from the blood into the tissue or the interstitial fluid outside of the blood vessel. Right, okay, so we'll start off with the bigger picture of the circulatory system. So we'll put the heart right at the centre here. So here is our picture of the heart. Okay, so this is the right atrium here, then the right ventricle, giving off the pulmonary trunk there, then the left ventricle over here, the left atrium here, okay, and then the great big aorta coming off here. Okay, right, let's draw the aorta arching over, and I'm not going to draw an anatomical diagram, don't worry, I just feel like drawing the aorta at least. Okay, so here comes the aortic arch, and then the descending aorta down here. And then what happens is the aorta gives off loads of branches, so it will give off arteries. So here we have another big artery, and the aorta is an artery, it's the biggest artery within the body. Okay, so you start off with arteries, so this is an artery branching off the aorta. Okay, then what will happen is the artery will split off into loads of smaller little vessels known as arterioles. So we'll show these. Oops, what am I doing? Putting an end to it, that's not good. Right, okay. So we'll show these little ones here. So these are arterioles, and they shouldn't have ends. I've just badly drawn it. Okay, so these are arterioles. And then if we look at one of these arterioles, so we'll look at the only one that I haven't ruined by putting an end to it. Okay, so this is going to branch off into loads of even tinier blood vessels, and I won't even give them a thickness, I'll just give them a line. Okay, and these smaller blood vessels here, these smallest ones, these are capillaries. Okay, and then what's going to happen is the capillaries will all reassemble into a little blood vessel known as a venule here. Okay, so here they come, all assembling back up into this venule here. Okay, so this is a venule, and then many venules will come together, okay, to make a vein, and this will then drain into one of the great veins. Okay, so these littler blood vessels here, these are capillaries. Okay, so capillaries, so these are the stars of the show. Right, then we have venules, these smaller blood vessels here, and I think I need to get some colour on this to improve it. Right, so we'll have the capillaries in turquoise, I think. So in turquoise here, these are the capillaries. Okay, you see they're improved markedly now, they've got some colour on them. Okay, so the capillaries are in turquoise. The venules, let's have the venules in orange, and obviously I'm not labelling these the colours, they will actually appear in the body. So the venules are in orange, okay. The arterioles we'll have in red, so they're actually given the colour they would appear. Okay, and then we'll have the arteries in this light green colour here. So, for instance, this is an artery, and of course the aorta is a massive great artery, but I don't want to waste my highlighter colouring in that massive great vessel. And then we'll have the vein here in blue. Okay, so the venules have drained into a vein, and then what will happen is many veins will drain into a great vein, and an example of a great vein is the inferior vena cava. So there's a massive great blood vessel draining into the bottom of the right atrium there, and it's known as the inferior vena cava, or the IVC for short. So this is this is meant to represent the inferior vena cava, which is an example of a great vein. And you have another example for a great vein, which will be the superior vena cava. Okay, and you have another great vein draining into the right atrium up here. So this is the right atrium, RA for short. And I'll label up superior vena cava as well, but I'll just label this as SVC for short, so superior vena cava. And of course, inferior vena cava would often be abbreviated to IVC. So both of these are examples of great veins. Okay. So, this is the picture of the circulatory system then. So we have the left ventricle over here pumping blood out of the heart into the aorta, and then, and I should label up the aorta. So this is the aorta. Okay. And then the aorta has arteries coming off it, which go into arterioles, and arterioles branch into capillaries. Capillaries then supply the peripheral tissues, and then you'll have the capillaries draining back into venules, and the venules will combine together to make bigger vessels, veins, and then the veins will drain into the great veins, such as the inferior vena cava and the superior vena cava. And I think just to complete the picture, we should then add in that it goes into the right atrium, then into the right ventricle, and then you have the pulmonary circulation over here. So this big blood vessel here, this is the pulmonary trunk, which will split into the two pulmonary arteries. Okay, one which will go to the right lung here, and one which will go to the left lung here. Okay, so the blood will go round the lungs, and then it will come back into the left atrium over here via the four pulmonary veins. Okay, so you have two pulmonary veins which are draining the left lung here, the left pulmonary veins, and then two veins which are draining the right lung over there, the right pulmonary veins. So these are the pulmonary veins. Okay, and I suppose I should label up the pulmonary trunk and the pulmonary arteries as well. So the great big vessel that comes straight out of the right ventricle here, this is the pulmonary trunk. So let me pull this down a little bit. Okay, so this is the pulmonary trunk, and then it splits into the two pulmonary arteries. Okay, so it splits into the right pulmonary artery, which supplies the right lung, which I'll just abbreviate to RPA, right pulmonary artery, and then the left pulmonary artery, which supplies the left lung. Okay, right, so that's our bigger picture of the circulatory system. So now we know what capillaries are. They're these things right in the middle, basically, and these are where you actually deliver, you actually deliver nutrients to tissue, basically. The arteries, you're not actually getting any nutrients being delivered to tissues in the arteries. The arteries and the arterioles are just vessels which supply the capillaries. The capillaries are where the actual exchange of nutrients and waste products is going to occur. So nutrients are going to leave the blood into the tissue. Okay, so nutrients will come out from the capillaries. So let me squeeze this in here. Nutrients. And then waste products will go back into the capillaries and be taken away from the tissue, and that's obviously the function of the circulatory system. Okay, to try and maintain the internal medium. Okay, right. So now that we've seen the bigger picture of the capillaries, let's now look at the different types of capillaries. And you have three major separate types of capillaries. Now we'll start off with the first type of capillary. So the first type of capillary is the one, if you were actually going to draw a capillary, usually people would draw this first type because it's the most common. Okay, so the first type of capillary is what's known as a continuous capillary. Okay, right, so let's draw a picture of this continuous capillary. Now, capillaries are tiny little blood vessels, but in essence, they are still a tube. So that's a good starting point for drawing a capillary, is to draw a tube. Okay, so here we have a tube, a nice cylindrical tube. Okay, right, so they are tiny little tubes, though. They are really, really tiny. Pretty much a single red blood cell will fill the entire tube, pretty much. So these are really, really narrow little tubes that just have a tiny little flow of blood through them. So their walls, therefore, pretty much consist of a single cell. So let me show this. So here we have this cell that's making up pretty much the entire wall of the capillary. So here's the nucleus of the cell in this little extension that I've drawn there. Okay, and then what will happen is this cell might, continue on, and it might make up this portion of the wall here. So this might be this one cell here. Okay, and then you might have another endothelial cell here, let's say, and we'll have its nucleus there. Okay, and another endothelial cell here. So by the way, these types of cells that are lining the blood vessel, these are known as endothelial cells. Okay, so they're very thin, they're a squamous cell, they're very flat, but they sort of span out. They're often compared to fried eggs because they have the yolk is the equivalent of the nucleus here, and then this sort of mass of cytoplasm that's just sort of really flat is the equivalent of the egg white. Okay, so these are often compared to fried eggs. These are endothelial cells. Okay, right. Literally, you have one cell spanning, making up this entire circumference of this blood vessel at once. So you can understand how tiny these blood vessels are. They can literally fit one red blood cell in through this lumen here. Okay, so here is the lumen. I'll highlight the lumen up in pink. Okay, so this is the bit where you've actually got the tube running through. Okay, right. So, some more things to discuss then. The junctions between the endothelial cells. So we can see here that we've got two endothelial cells meeting. Now, often these junctions will be held together very, very tightly, especially in continuous capillaries. So if we now draw another picture. Okay, we're going to need quite a few pictures to get a proper understanding of this. So here's our picture where we're looking from the side. Now let's draw a cross-section picture. So let's draw as though we're standing here and looking at this portion here. So we've got this sort of circle now. So it is a circle. It looks like an oval there because I'm trying to draw a 3D picture. Okay, and then we've got this one endothelial cell wrapping itself all the way around the circumference of this tube. But then, of course, there will be a point where it has its processes end and they join there. Okay? Right. So, what sort of a junction do you have here then? So to understand that, we need yet another picture. So we need a picture as though we're standing in the tube and looking at the at the endothelial cells from within the tube. Okay, so if you look from within the tube, what you generally see is you'll have endothelial cells, massive, great endothelial cells, okay? But then they'll have points where they need to join to another endothelial cell. So here's one endothelial cell, and now you can see the fried egg comparison coming up, okay? And then here's another endothelial cell. Right, so we need some way of joining these two together, and in some cases they don't actually have this tight adhesion to one another, and we'll discuss that in a moment. Okay, so what you generally have is, if I just draw one final one, so I have one final one sort of here to get the message across. Okay, so usually what you have is a very tight protein meshwork in the gap between the cells, okay? So in this sort of space between neighbouring cells, you have a very tight meshwork sort of going into every little nook and cranny, basically, and forming a seal between the two cells. Now, this is often compared to a six-pack of beer. So, if you've ever bought a six-pack of beer, not one that's in a cardboard box, but one that's free, where you can actually see the beer cans that you're buying. Okay, so just to draw a little picture of this. So if you've got a beer can here, so here's the can, okay, and let's say here's another can, and because I don't want to have to draw 6 cans, we'll say it's a 2-pack of beer, but with the same sort of packaging. Now, if you want the two cans to have to be sold together, but you don't want to have to put them in a plastic box together, the beer companies have come up with rather an ingenious solution to this. What they do is they make a thin sort of 2D slice of plastic, and this slice of plastic will have holes in it where the beer cans are. Okay, and these holes will be really tightly wrapped around the beer cans. So basically, you can pick up this piece of plastic, and the beer cans will stay in the piece of plastic, and they're both attached in there. Okay, but then all you need to do if you want to get the beer out is then cut the plastic, or often people just stretch it, and then the beer cans fall out because the holes become wider. Okay, so tight junctions are often compared to this sort of packaging because the cells are like the beer cans, and then the plastic is like the tight junction, wrapping around really tightly the endothelial cells, okay, and forming a very tight seal. I mean, you cannot get your finger between between the apical face of the beer can and the basolateral face of the beer can. You can't get it past there, so it really is a very tight junction. Okay, and that's what tight junctions are like. Now, between most endothelial cells, you will have these tight junctions. So I'll highlight these in blue. So often you will have them. However, there will be places where tight junctions are absent. So for instance, let's say this spot here between these two endothelial cells does not have a tight junction there, okay? Now this means that the cells are still very sort of tightly pressed against each other, but there isn't actually this thick protein structure that is stopping things from being able to move through that gap. So there is a tiny little gap between the two endothelial cells, and that gap between the two endothelial cells is what's known as an intercellular cleft, okay? And intercellular means between separate cells, and a cleft just means a hole. So an intercellular cleft. So it's a gap between the two endothelial cells. Now, do not fear. This is a very, very small gap. So things can move through, but not big things. Small molecules can move through this gap. Okay? The important thing to understand is that cells are not going to be able to move through this gap, so blood cells aren't going to be able to move through this gap, and also, very importantly, the coagulation factors are not going to be able to move through this gap, because if the coagulation factors moved through this gap, you'd get coagulation beginning, you'd get fibrinogen being converted into fibrin, and then fibrin being assembled into fibrin strands, and you don't want that, because, you know, this is supposed to be there. You don't want this being plugged in with a hemostatic plug. Okay, so cells can't move through there, so as an extension, platelets can't move through there either, and the coagulation factors can't move through there. So that both of those things, platelets not being able to move through there and coagulation factors not being able to move through there, means that you're not going to get a hemostatic plug being produced in this gap here, which would be a disaster. Okay, so we've got these intercellular clefts between certain of the cells, and then a lot of the cell junctions will just be tight junctions. So mainly in continuous capillaries you have tight junctions. Occasionally you'll have some intercellular clefts, but the important thing is that you have very few of these intercellular clefts. So this is why they're called continuous capillaries, because it forms a nice continuous confluent layer of endothelial cells, all nicely adhered together, and with a few intercellular clefts here and there. Okay, so now just to complete our discussion of continuous capillaries, I need to tell you about some more layers. So overall, so far I've just told you about the endothelial cells, okay, but there is actually a layer outside of the endothelial cells, okay, which I'm showing now in turquoise. So the endothelial cells are sitting on something, basically. They're attached to something themselves, because you have to remember, these cells are just goo, basically. What's holding them in this structure? Well, basically they are attached to a very strong protein meshwork. So this is kind of like it's kind of like the infrastructure of the capillary. It's what's holding the capillary in its shape, basically. Okay, so this protein meshwork is what's known as the basement membrane, and it mainly consists of the protein collagen, different types of collagen, but overall collagen, we'll say. It also has other proteins in it, such as fibrillin and laminins, okay, but mainly collagen. So this is a protein meshwork that the endothelial cells are attached to, and it's what keeps them in their correct shape and keeps the blood vessel in its correct shape. Okay, one final thing to add also, and this will become important when we come on to discuss fenestrated capillaries, is that these endothelial cells have on them many polysaccharides, basically. On their apical membranes, they have polysaccharides. Okay, and these polysaccharides are attached to proteins which are in the apical membrane of the cells, and for that reason these are often referred to as proteoglycans. Okay, so a glycan just means a polysaccharide, basically. It means a polymer of glucose, strictly, but these are actually slightly more varied than that. They have many different modified versions of glucose in them. Okay, and the proteo means that it's attached to proteins, so it's a polysaccharide that's attached to proteins in the apical membrane. Now, this layer of polysaccharides over the apical surface of the endothelial cells has a name. Okay, so it's made of proteoglycans, but that's not its name. Its name is the glycocalyx, or the glycocalyx. Okay, so the glycocalyx is this layer of polysaccharide that is over the apical surface of the endothelial cells. So an important example of one of these proteoglycans that's over the surface of the endothelial cells is heparan sulfate. Okay, and heparan sulfate plays important roles in preventing coagulation from occurring in in healthy blood vessels. Okay, so these continuous capillaries then, they're called continuous capillaries because they have these tight junctions making the endothelium one nice, confluent, continuous layer. Okay, they have very few intercellular clefts, and you find them all over the place, basically. So you'll find these in your fingers, you'll find them in your brain, you'll find them, I think, in the gonads, so they're all over the place in the body, basically. Okay, so in the next video what we'll do is move on to fenestrated capillaries.